Good morning, everybody. I'm pleased to present Dr. Kevin Gardner. Dr. Kevin Gardner held several positions at the NCI. He was a senior investigator at the genetics branch, and he was also the acting scientific director at the National Institute on Minority Health and Health Disparities. Presently, he's the senior vice chair of the Pathology and Cell Biology Department at Columbia University Medical Center. He's also a professor in pathology and cell biology at Columbia University Medical Center and the director of the Digital and Computational Pathology at Columbia University Medical Center. I serendipitously met Dr. Gardner at a visit at the NCI, and now I'm very happy to count with Dr. Gardner as one of our collaborators in an ongoing project. So today, I leave Dr. Gardner to talk about the race and molecular biology of breast cancer, a pathologist's view from the bench to the bedside. Thank you.
Okay, well, thank you. Start off by thanking Maria for that introduction. I'm a clinically trained pathologist that studies chromatin-based mechanisms of transcriptional regulation. That sounds like a lot, but I do what everyone in this room does. I seek to understand disease in terms of its most basic molecules and interactions. Then we try to assemble those interactions and molecules into patterns that represent pathways and programs of cellular phenotype, function, and physiology. And the overall goal is to improve disease diagnosis, disease treatment, and disease prevention. Okay? So I'm going to organize this talk. Well, first I designed this talk to be for a general audience, but I have been reminded on several occasions by my wife, who is an artist, that I don't know what a general audience is. But bear with me. Okay. So I'll divide this talk into these 5 parts: a brief review, very brief review of breast cancer epidemiology and race, a basic overview. We won't get too deep into the weeds about perspectives on epigenetic regulation. And then I'll discuss 2 epigenetic regulatory proteins or genes that we've been studying in the lab. And I picked these 2 because they illustrate a project where we went from the bench to the bedside, and another project where we actually went from the bedside back to the bench. Okay. Then I'll review the use of digital pathology and artificial intelligence in bench to bedside approaches, and then how we use that to illustrate or elucidate aspects that improve diagnosis, but also teach us new functions about proteins we didn't really realize previously. And we'll talk a little bit at the end about the new biomarker discovery using artificial intelligence. Also, if time permits, I'll make a few comments about race and how race is a social construct, and also, if possible, make some comments about collaborations and how to do them, and how do you do multidisciplinary collaborations where you're really you're looking at each other from different perspectives, and you're trying to come up with a common language. Okay. All right. I have no disclosures. All right. Last year there were over 260,000 new cases of invasive breast cancer in the United States, with estimated over 40,000 deaths. The effects, the influence, the distribution of cancer is unevenly spread across different populations, where in women of African descent have a nearly 40% increased mortality by comparison to women of European descent. The reasons for this difference is multifactorial. They involve a spectrum of different aspects or different parameters in health determinants: treatment, screening, socioeconomic status, social behavioral aspects, comorbid conditions, and stage of presentations, which are all interconnected. Stage of presentation is linked to screening. Treatment is linked to stage of presentation. Today, we're going to focus a lot more on tumor biology. Okay. All right. So a little bit about the approach that we use in chromatin biology. We begin essentially with trying to understand the basic elements of interactions, the basic processes, and then we try to translate what we learn into understanding mechanism. And then we try to, if we can, link those mechanisms to something that's important and useful in understanding health, understanding disease, increasing treatment, improving treatment, and increasing prevention. Okay? So we begin with understanding basic mechanisms, then we try to translate. Okay? All right, so the major phenotypic drivers of cancer are basically two. There's gene mutation. This is an irreversible process. And then there's epigenetic regulation. Unlike gene mutation, epigenetic regulation has many different parts or pieces, very different. Unlike gene mutation, epigenetic regulation is reversible. Okay? In addition, epigenetic regulation can be influenced by factors or properties in the environment. Okay? Okay. All right. I think the best way to try to think about understanding epigenetic regulation is to think about chromatin packing. Okay, so let's start a narrative. Okay, each one of our cells in our body has 2 meters of DNA, 2 meters, and we pack that into a 6 to 12 micron nucleus. That's a pretty impressive compaction ratio. That's a 10,000-fold compaction ratio. Okay, that's like taking a 100-page book and then folding it up into the size of a period. So that's a lot of information that's packaged. Now, that folding of the genome creates issues, creates problems, because in the nucleus you have to accomplish all these different biological processes: DNA repair, DNA replication, transcription. And so the cell has evolved many different ways of opening and closing the chromatin. And that is the crux of epigenetic regulation. Opening and closing the chromatin so the molecular machinery in the cell can get access to the DNA and do what it needs to get done. Gene expression, gene repair, gene replication. Okay? And the properties of the molecules that are important to that process are referred to as histone changers, things that essentially change the basic components of the chromatin, the histone. And some are referred to as histone writers because they make covalent modifications to the histone. Others are referred to as histone erasers because they remove those covalent modifications. And others are referred to as histone readers because they bind to those modifications and they can home in other molecules, other factors that actually use ATP to help open up the chromatin. All of what I described is under that whole realm of epigenetic regulation. And so the key factors are the modifications to chromatin. So opening it up and close it and moving it around using energy. Okay, so we'll begin with one protein that we've been studying in our laboratory, a protein called C-terminal binding protein. This is a dimeric protein, 2 family members. It can form homodimers and heterodimers. Interestingly, it's highly expressed in developing tissue. It plays a major role in epigenetic regulation of cell fate, cell phenotype. And it's elevated in a variety of different cancers, including colon, breast, prostate, and ovary. Interestingly, its activity is dependent on NADH, and we'll get back to why that might be important later on. So it forms dimers and heterodimers. So in the nucleus, it can form complexes with other chromatin targeting complexes, chromatin binding proteins or proteins that bind to chromatin bound proteins. The other aspect of this protein is that it can also bind to chromatin modifying proteins, the readers, the writers, the erasers. So in this way, it can essentially home different chromatin modifying complexes to all different landscapes across the nucleus. Okay, and because its modification or its activity is dependent on NADH, it can do that in an energy-dependent or metabolically driven fashion. So, in this way, we look at CBP as a metabolic switch that can actually change or reshape the epigenetic landscape within the nucleus. Okay, so we're not going to get too deep into the weeds here, but what we did to understand CBP was to use chromatin immunoprecipitation, a way where we can actually track where the protein binds throughout the nucleus in different cells under different conditions, and we can actually track that by looking at these different peaks that it forms, and then we ask where in the genome or what genes are they bound to. Then we can start to extrapolate how they function. Some of the things that we began to learn was that their CtBP is bound at many promoters and enhancers, and it has specific sets of proteins that are protein sequences or DNA sequences that bind specific protein DNA binding proteins that's enriched at. Okay. And so we were able to do genome-wide mapping to get a sense of how CBP binds throughout the nucleus in relation to other genomic regulators that are important in breast cancer, including BrCa, including P300, Pol2, and estrogen receptor. And we found that there was a significant overlap between CBP and many of these complexes that have a significant role in controlling estrogen receptor-dependent transcriptional control. Okay. And after looking at a variety of these different complexes, we began to see that CBP may be actually part of a much larger complex that's been now referred to, it's only about 2 years old, as a mega transcriptional complex that plays a significant role in controlling estrogen receptor-dependent gene expression, but also it has been implicated in pathways that lead to endocrine resistance in breast cancer. Okay, so we've done a variety of characterizations. I'm not going to really go much into detail, but that involves looking at the biology of this protein. We can overexpress it, we can deplete its expression, and we can ask whether or not it has an effect on cellular behavior. Does it increase cellular aggressiveness? Does it increase invasion? Does it increase motility? And it does. We were surprised to find also that not only does it respond to changes in metabolic status, like Warburg phenomena, but it also can drive changes in metabolic status. It can drive glycolysis. We had 2 different assays on the left. We did this in a triple negative cell line, and we did a metabolomic study. We found that high levels of CBP could actually drive a glycolytic process. We went into an estrogen receptor positive cell line. We also found through specific measurements like the Seahorse assay, we found the same thing, that CBP drives glycolysis. Okay, we've done genome-wide expression analysis by RNA-seq, and we find that when cells are depleted or deleted of CDBP, one of the CDB members here was CDP1, it has a signature that suggests it has a major role in epigenetic regulation of phenotypic states, phenotypic plasticity, as well as have a major effect on metabolism. We've also been working with collaborators in developing small molecules that can actually inhibit CBP. So part of our research approach or our research program is to look for ways to repress the activity of CBP, move into animal models, see what kind of combinatorial therapeutic approaches that we can come up with. And so in analysis, in a nutshell, we think that CBP is this metabolic switch that can, in a bidirectional way, influence epigenetic regulation, but also drive differences or changes in metabolism that also acts in a feed-forward fashion to influence epigenetic regulation. Okay, so this is the bench to bedside project. Where is the bedside part? Now, most of what I just described was done until the last 25%. Most of that was done while I was at NCI, and some of you may not know, NCI does not really have a breast cancer program, and so we're left saying, well, we made all these measurements, and we're pretty sure that CDBP has something to do with breast cancer, right? But how far could we get to prove that? And so a former attending of mine was the chair of East Carolina University's pathology department. And so I called him up and I said, well, we think this protein is important in breast cancer, but we have no way of proving it. We need patient samples. And he said, well, we have a lot of patient samples, but we're a teaching hospital. We don't have, the manpower to actually do the pathology. If you come down and do the pathology yourself, we can probably work that out. And I said, well, good thing I'm a pathologist. I have a car and a microscope. And so we actually drove down to Greenville, North Carolina. And when I arrived there, we had paid them a stocking fee. And so we asked them to pull all breast cancer cases, stage 0, from 1999 to 2011. And they did that. And so they ushered us in to the second floor where they had histology, and they pointed us to this room, walked in this room, and it was like, maybe 270 square foot room with shelves that went to the ceiling, and they had pulled 20,000 slides. And so we had our work cut ahead of us, and we just went through the slides, labeled them. And the way we did this in this type of collaboration is that I brought a flatbed scanner. My staff scientists kept the records. I read the slide anything we wanted that had tumor on it, we scanned. And then we handed the scan of the slides back to histology, say, we will pay you now a stocking fee to pull those blocks. Now, in the interim, we had to sign a lot of IRB agreements, et cetera, et cetera. And I must tell you that in the middle of this, I'd gone through 20,000 slides. We had, requisitioned about 2,500 blocks. We were starting to get the blocks. They were arriving at NIH. We were starting to go through everything. And then surgical path numbers became protected health information. That was a nightmare. Okay, so I just want to point that's we got through it, but that was a major nightmare because we had to redo and re-anonymize everything. Okay, but we got through it. Now, to say something about where we're getting the samples and why we're getting these samples here, the region that's served by East Carolina University Hospital, which was now called Vidant Hospital. It's a designated health disparities area. Cancer health disparity has been designated as so for over 20 years. The population is 1.4 million. It's rural, 97 folks per square mile. I live in New York City. That's just amazing. Poverty is 25% is below the poverty level. It's a very diverse population. 40 to 50% African ancestry, and it's in the South a very high level of obesity. Okay. More interestingly, single healthcare system, and 80% of all the surgeries are done at this hospital. So any data that we could extrapolate from this would be almost population-based type of data. Okay. All right. So we went to 20,000 samples. That ended up being about 2,500 blocks. They ended up being about 900 cases that are, tumor that we could actually look at. So the first thing we thought of doing was getting them onto TMAs. Okay, and then there were a few that we could, through the IRB, we could get some genomic sequence on because they were deceased and we didn't have to get consent. Okay, so we did some panel sequencing, about 147 of them, and then we did RNA-seq on that same sample. And then the next thing was the data integration, get information from the charts. Get as much as we can from patient phenotypes. And then the most important thing that, we were very excited, and then we realized the sober reality was that now we got to look at it. Okay. So, some of the things I'm going to talk about are things we did out of necessity. Okay. And so the first thing we started to do out of necessity is think about digital pathology. Okay. Now, digital pathology is a very broad term. The actual use of artificial intelligence is a very broad term. The best description is the use of machine learning that you can train and supervise programs to do to help you make classifications. That's it. Classifications can be of any type, morphological classifications, subcellular classifications. And so we started out in essentially two ways. We began first by just pathologists annotating the tumor, but then we could use machine learning to detect nuclei, detect cytoplasm, and detect membrane. And we could actually get a digital score of the staining for any antigen we wanted and dictate or indicate or classify whether it was membrane, nuclear, or cytoplasmic. That then allowed us to get a continuous score. Now, since then, we've been getting a little more sophisticated. And we're actually teaching the program to actually find the tumor for us, and then it does a secondary analysis. So you can actually have 900 cases, and you don't go blind, doing counts and getting an IHC score or histo score. So in pathology, we use a histo score that represents the heterogeneity of the sample, the percentage of cells that are 3+, percentage are 2+, percentage are 1+. You basically get a continuous score from 0 to 300. That allows you to do a lot because you now have a continuous score. You can do multivariate analysis. It's really an enabling technology. And this is not deep learning where we're asking the computer to replace a pathologist, which by the way, is not going to happen anytime soon. They may replace radiologists, but what we feel that the canary in the mine is the radiologist. If they go, then we're next. Okay, but that's not going to happen anytime soon. The day that they can put a computer on the witness stand and talk about a suit, then we won't have to worry about it. That's not going to happen. Artificial intelligence gives you sensitivity. The pathologist domain is specificity. Okay, it's a workflow advantage. It'll speed up diagnosis. But if a case comes back negative, the pathologist's job is to make sure it really is negative. Okay. All right, so this is an example of something we just recently published where we actually asked basic questions about the continuous score, and we can actually ask questions about the relationships between different antigens. We can ask questions about the relationship between different antigens, and we can ask quantitative information about continuous patient parameters. Okay? And here we basically were able to show that there actually are racial differences in the predictive value of different standard antigens. In particular for estrogen receptor. Estrogen receptor is a much better predictor of good survival based on genetic ancestry, okay, or based on race. And we'll talk about the difference between ancestry and race and how I got into trouble trying to make it easier later on, okay? We also found that, genes that are master regulators, genes that form complexes with CDP, are significantly, differentially predictive of survival based on race. Okay, so let's get back to CTBP. So this is an experiment where we essentially asked, is it true? If we looked at the nuclear localization of CTBP, was there any correlation between CTBP levels and breast cancer? And here we show there's high levels of nuclear CTBP is associated with poor survival. High levels of nuclear CTBP2 is even more predictive of poor survival. And it really is subcellular specific. If it's more in the cytoplasm than the nucleus, it's a favorable prognostic factor. If it's more in the cytoplasm than the nucleus, again, for CTBP2, it's a favorable prognostic factor. So we can really tie the presence of this protein to where it is in the cell and its total amounts. Okay? We could not do this and get this type of data without using machine learning or artificial intelligence. Okay. What was a surprise to us that there was actually, CTBP1 and CTBP2 were differentially predictive of survival from triple-negative breast cancer based on race. And that has become a major focus of some of the work we're doing now. Okay, so our final conclusion here was that we started out with a protein, we extrapolated its role based on understanding molecular interactions and linking those to pathways, and then come up with the conclusion that this protein may have something to do with breast cancer, and we have drugs that may inhibit its activity. Okay, so that's the one bench to bedside story. Okay, now I have a new story that comes out of collaborations across the country with Clayton Yates at Tuskegee, with Juliet Daniels at McMaster's in Canada, where we're looking at this protein called Kaiso. Kaiso has been defined as typically a nuclear protein that can have a role as a repressor or an activator in the nucleus and can be influenced by epigenetic changes, including methylation of histones and methylations of DNA. More importantly, Kaiso can be found in both the nucleus and the cytoplasm, just like CBP, but there's something different about this. Now, we began this collaboration with Juliet and Clayton because they had found that in women of African ancestry, and basically this was mainly looking at Juliet's cohort from Barbados, that there's higher levels of Kaiso in the nucleus in women of African ancestry, particularly Nigerian, and that it was much more highly predictive of worse breast cancer survival, again, based on race in women of African descent, particularly Nigerian. Well, it's okay. We started as a collaborator, so let's look at we have a larger cohort, and let's look at our cases that we had annotated. Essentially, the cohort now is about 900. It's taken a while to annotate it, but we now have about over 9 years of median follow-up on 670 patients, and we're going to backfill in on the rest of those 900 in the next couple of years. But anyway, to make a long story short, we started staining the tissues, and we said, hey, there's staining in both the nucleus and the cytoplasm. And they said, yeah, well, it's the nuclear part that matters. Well, we had a digital way of looking at and making the correlations. And so we digitally asked, what is the scoring in the nucleus versus the scoring in the cytoplasm? And we found that what Juliet and Clayton were right. Nuclear Kaiso was predictive of poor survival, but cytoplasmic Kaiso was much more predictive of poor survival. So we started to ask general questions about, is there a difference in the activity, the associations between cells that have high nuclear Kaiso versus cells that have high cytoplasmic Kaiso. We did correlations and distributions, and we found that it was not the same story as CTBP1 versus CTBP2. It wasn't inverse. If you had high Kaiso in the nucleus, you could also have high Kaiso in the cytoplasm. And so there seemed to be something very different about that. So we started to ask these general questions where we had continuous scoring for different biomarkers, and we asked about those relationships. And what we found was that we could use unsupervised scoring and get distribution of protein levels, red as being high, blue being low, and we could then correlate that with the patient sample data, including subtype data. And we found that there was a significant correlation of the cytoplasmic CBP with triple negative breast cancer. We also saw that there's this one antigen that has been used to look at autophagy related events, secretory events, that was also highly correlated with Kaiso. So those are very interesting. Again, this is a correlation we're making purely from looking at patient samples. Now, autophagy is a cell stress response pathway that after transformation of the tumor can lead to resistance to stress in the environment, resistant to hypoxia, resistant to drug therapy. We measured our antigen. The antigen for autophagy is LC3. We, just like Kaiso, LC3 predicted poor survival. It was also highly elevated in triple negative breast cancer, as you can see here, right? And there was a slight difference based on race. All right. What was also very interesting is that using just two antigens, Kaiso in the cytoplasm, not Kaiso in the nucleus, which is down here, Kaiso in the cytoplasm, and this LC3 marker, this secretory autophagy marker, we could actually stratify triple negative breast cancer into different survival groups. So patients that had intermediate or high levels of Kaiso and intermediate or high levels of LC3A, this autophagy-related molecule, had poor survival. That's B1, B2. But if you had low levels of LC3 and intermediate to low levels of Kaiso, you had survival that was up here. So it's stratified survival. Okay? So we became very interested in how to figure out what this protein does. So in our next step, we took advantage of a couple of resources that we had from this cohort. One resource is that we had RNA-seq data. Okay? So using RNA-seq data, that gives us a way to get a sense of the actual transcriptional state of the cells. Okay? And in combination with the RNA-seq data, stratifications of the patients by whether or not they had high or low Kaiso in the cytoplasm versus the nucleus, or high or low LC3, we could actually ask questions about the actual transcriptional state of the cells using gene set enrichment analysis. Now, gene set enrichment analysis is a way of using curated data sets that have been used to essentially define the transcriptional or gene expression state of the cell based on the enrichment, the differential enrichment of different genes. Okay, so what we did was take essentially 4 different categories or 4 different stratifications of the patients and use that RNA-seq data. We stratified them by high versus low nuclear Kaiso, high versus low cytoplasmic Kaiso, high versus low total Kaiso, and then we looked at RNA as well. And then this analysis is done by Sandeep here, who's in the audience. It was just a mind-boggling thing. He looked at over 17,000 different gene sets to see which ones show the most enrichment, and the color here is the enrichment score here for these different categories. It turned out that the most useful, that most annotated data set was the GL data set. Okay, and that itself contained about over 9,000, nearly 10,000 gene sets. And then we looked to see what gene sets were overlapped for at least 2 in any of the 4 categories. Any of the 4 categories, meaning categories of stratification of the patient's gene expression based on how we quantitated the nuclear versus cytoplasmic expression, high versus low. Everything was above and below the median. Okay. And when we did that, we found that there's about 200 or so genes that are at least present in 2 of the categories, and we started to search for things that might be important that are actually enriched. And one was significant enrichment for stress and metabolic pathways. Another was actually enriched for autophagy. So we were looking for a needle in a haystack, and we kind of found it. All right. And then we actually just wanted to look at the statistics of the relative enrichment. We found that if you look at stratification by IHC scoring of nuclear versus cytoplasmic versus total versus RNA, that the highest enrichment or most significant enrichment of these autophagy and gene secretory terms was only in those patients that had high levels of LC3 or high levels of Kaiso in the cytoplasm. Okay. So we're now beginning to our journey from the bedside to the bench in this story. Okay? So the patient data told us that there's a link between Kaiso and autophagy or autophagy-related factors. So we took a triple-negative breast cancer cell line and depleted of Kaiso and asked what by, looking at gene set enrichment analysis and also looking at a curated data set of autophagy genes. And we found that of the 500 or so autophagy genes, about 142 of them were altered in cells that had altered expression of Kaiso. And there's a significant enrichment, once again, for autophagy terms in the cell line, not in the patients. So we went from the patients to Kaiso to autophagy or autophagy-related factors, autophagy-related antigens. We could start to make that connection. Then we did a physical assay. One of the physical assays for looking at autophagy or autophagy-related factors is that when it's active, it forms these punctate structures, the autophagolysosomes. And we show here that in the wild type, you can see these little, the arrows are pointing to a little punctate dot. The knockouts with 3 different hairpins didn't make autophagolysosomes. So we could actually disrupt autophagy-related processes by decreasing the levels of Kaiso. And then we did fluorescence, and we could see there's a significant, and this is a triple negative, this is a ER positive cell line and triple negative cell line. We could see that there's a significant co-localization between CBP between Kaiso and LC3 in both the cytoplasm and the nucleus. So there's a lot of things we began to learn about this, and this is simply, again, teaching us by looking at patient samples and understanding interactions by patient samples. Now, so we went back to the patient samples and asked, can we see any type of co-localization of Kaiso with LC3? And this is DAPI stain on nuclei. That's Kaiso staining, and it's mainly in the cytoplasm. This is the LC3AB staining. That's actually a mixture of in the cytoplasm and the nucleus. This is the overlay. This is kind of a blow up of it, and it's a significant interaction between Kaiso and LC3AB, not only in the tumor cells, but we also see interactions in the stroma that we want to look at in the future. That was a triple negative. This is a luminal B cell line, and we see similar interactions, but we always see a higher level of co-localization in triple negative breast cancer. Okay, this next figure is just a mapping of the co-localization, again showing it's a significant level of co-localization. Green is just Kaiso alone, red is just LC3AB alone, and yellow is the co-localized signal. TNBC, luminal B, and so this is a quantitative co-localization. Okay, so now we had a new story that we're beginning to pursue. Now, there's in some of the gene expression data that we were looking at, both the patients and in the cell lines, we noticed that there seemed to be an immunosuppressed signature, simply by looking at gene expression enrichment profiles by gene set enrichment analysis in patient samples, but also in looking at our cell line data. If we depress Kaiso levels, a significant number of class 1 and class 2 molecules started getting upregulated in those cells. So we started to ask a general question about, is there an immunosuppressive aspect of Kaiso? Can Kaiso contribute to immune suppression? And the first place we want to look is in the tumor microenvironment. Okay, so this is a study where we basically stain our tumor microarray for CD8, for CD68, and in this case, PD-L1, and pancytokeratin to classify tumor versus stroma. And then we took this one step further. Okay, so on the left is just the staining pattern. On the right, essentially, we actually constructed a 2-dimensional Cartesian plane where every single nucleus on that tissue core was mapped and phenotyped. And then here we have the mapping and phenotype of every single nucleus. And then we ask, again, using machine learning, this is only done by artificial intelligence, what is the distance between each different phenotype throughout the whole tissue? And can we see any differences in the distribution? And moreover, since we have a new dimension of data from our Kaiso scoring, can we relate that to Kaiso scoring? In fact, can we relate that to any type of scoring? Can we relate that to race? And so this is our first pass at this. And this is a graph where on the x-axis is the distance between the 2 different nuclei. Okay? The average distribution based on their immunophenotype. In this case, we're just looking at CD8 cells that are PD-L1 positive, and what is their nearness or closeness to tumor cells, and how does that change in response to the levels of antigen. And here we show that if we look at cytoplasmic Kaiso, there's a high increase in the proximity of immunosuppressed T cells near tumor when there's high levels of Kaiso. Similar results, not as significant, but similar results with LC3. We do not see that with nuclear Kaiso, and we actually see a difference based on race. Another thing we're beginning to take a closer look at. Similarly, we looked at CD68 cells that were PD-L1 positive. We see a similar relationship, not as strong as CD8 PD-L1 positive. We can also do the converse. We can ask about all CD8 cells and what is the spectrum of nearness of all CD8 cells to tumors that are PD-L1 positive, and we see the same relationship. We also see the same relationship based on race, particularly with CD8, and a similar trend, in some cases significant, in some cases not significant based on race, or CD68 near PD-L1 positive tumor. All right, so the conclusion from this is that the levels of Kaiso in the cytoplasm correlates with autophagy-related factors or autophagy-related events, which in many cases are secretory, and that itself correlates with an immune-suppressed tumor microenvironment. Okay, and if we do the controls, if we just look at TILs, not the same. We're not looking at just total number of cells that are infiltrating the tissue. It's an immune suppressive profile that we found that is actually related or correlated with the expression of a specific set of tumor antigens in a tumor antigen related pathway, including autophagy, not necessarily autophagy, because autophagy is linked to all kinds of things that are secretory that in essence influence the tumor microenvironment. And again, our controls, we don't see that. Not CD68, not CD8. It's not a TIL-based phenomenon. We're not predicting TILs, we're predicting an immune suppressive environment. Okay, so that was on the TMA, and now we're beginning collaborations. Now that was fluorescent. You might all guess that that's not cheap, okay? And that's why we did it on TMA, and we got some really reasonable data, but we now really have to do this on larger real estate, okay? There have been several studies in using TILs, just looking at TILs, counting cells, counting a lymphohistocytic infiltrate, and it's been shown that both TMAs and whole sections give you predictive value. They don't agree, but they are both predictive of survival or predictive of our hormone status. We're not going to do that we can't do it on a whole slide. So we work with a team that has worked out this stain using chromogens. Okay, so you use 5 or 6 different chromogens, and you look, well, I'm just showing you a little bit, but this is just like, it looks like hell. This just looks terrible. You can't read it, but a computer can. Okay, and so we're working with a group at Stony Brook to use deep learning to look at proximity between tumor and immune cells on the whole slide sections. Okay, future studies include us looking more at topology, and we're using real deep learning now to look at the relationship between where we see Kaiso highly expressed and where we see it maybe in the stroma, or correlating that to expression of LC3 and Kaiso in the tumor versus what we see in the stroma. That's a topological equation. And so we can use deep learning to look at a feature of topology that's referred to as persistent homology. That's over horizon. All right, now, let's get back to race. I got schooled by a reviewer. The paper got published because I was, we were referring to race as ancestry. So we were saying African ancestry versus, European ancestry. And of course, we knew it was based on self-reported, a binary self-report. And because we were looking at biology, and I'm explaining myself. I lost. Okay, he won. Okay, basically he was right. He actually said, look, you can't do that, even if you think it has a biological implication. Ancestry is a continuous variable. You shouldn't act and treat as if it's binary. And yeah, you're right. I think it was Rick Kittles. I don't know, but he was right. So I think it was Rick Kittles, actually. So, we had some data. We had some RNA-seq data, so we could actually do some ancestry just to see, what do we have? It's too small of a data set. It was about 140 cases, but we could actually look at ancestry. And the cohort that we're looking at, those who self-identify as non-Hispanic white are 99.4% European. Those who self-identify as non-Hispanic black are mixed, 80% mixed. And so Then we went back to our data and we said, well, maybe there may be some differences that we couldn't pick up, and maybe due to heterogeneity. Let's look at some confidence intervals and see if we can see differences based on rice that Juliette Daniels saw and that Clayton Yates saw that we weren't seeing. And well, you know, we're kind of right. It turns out that cytoplasmic Kaiso is more predictive of poor survival based on race, and nuclear Kaiso is more predictive of poor survival in triple-negative breast cancer based on race, which was reported by our collaborators. So, you know, they pointed out one thing in a paper that we published with them a couple of years ago, where they were comparing a southeastern cohort to our Barbadian cohort, and I could see the dichotomy. And the issue was this is probably explained by admixture. Okay. All right. So I will begin one more narrative. Okay. This is a narrative you all know. Okay, but it helps us put things in perspective. 300,000 years ago, you know, man began in Africa. Okay, and then 230,000 years later, after being shaped by pathogen exposure, being molded by changes or shifts in nutrient availability, they migrated out of Africa into Europe, into Asia. Okay, and then 20 to 30,000 years later, some folks migrated across the Bering Strait and populated the Americas. So you basically had the four major superpopulations. You had African, Asian, European, and Native. Okay? And then there's this guy named Columbus, who floated over here, made a wrong turn, and ended up doing, causing all kinds of mischiefs. You had the slave trade, you had the conquistadors, and now you have an admixed population. Okay? And so it's still, for me, it becomes, it reminds me of a lot of different things. And it begins with some ironies. How do we define race? Race is a social construct. In most cases, a ridiculous social construct. How much time do we have left? Okay. And let me show you a few examples. Okay. Some of you people might recognize some of these folks. Okay. Does anybody recognize any of these guys? Okay. This is Rachel Dolezal. Rachel Dolezal was born 100% European ancestry, and she decided to relate more with a Black ethnicity. She was very effective. She went to Howard. She was the head of the NAACP in Tacoma, Washington. You remember that. Until it was found out that, her parents said, well, that's our daughter. and so then that led to a lot of trouble for her. I mean, things happened. She, this is bad, and, she was ostracized. And, I said, I feel for the sister. Okay? Because it wasn't until they found out that she was white that she learned out what it was like to be Black in the United States. Okay? This other guy is Ralph Taylor, an interesting person. The advent of DNA sequencing, ancestry DNA, has made all kinds of things. It's also caused interesting conversations at the dinner table, which I'll explain about one of the conversations at my dinner table. Well, this guy found out that he had maybe 6% Native American and 4% African ancestry. So he's been suing the government to get a minority businessman's law. Okay. Race is a ridiculous social construct. Okay. All right. Now, this is an interesting story. Okay. These folks are from a place in New York State, very close to the New Jersey border, called the Ramapo Mountains, in the Ramapo Mountains region. The place is called Hilburn, right? In 1943, Thurgood Marshall came to this town because he was challenging the Plessy versus Ferguson decree or act, whatever, stating that separate but equal was okay, right? And he came to this town because all the white folks were in the nice school, and all the folks of color were at a school that had no running water and no heat. Okay. So I show you this picture because that's my wife's mother right there. Okay. And I guess you can't see the pointer. Yeah, I guess you can't see the pointer. Can you see the pointer? Okay.
Showed up.
It showed up for me. Let me see if I can supplement that. Okay, that's my wife's mother. Okay, and I just want to point out that Thurgood Marshall came to her town in 1943 because she and this. Oh, I think it's freezing. Yeah, I actually did freeze. That's what. Yeah, because she and this strapping young colored boy could not go to school. So, race is just a completely ridiculous social concept. Now, finally, and then we still have some time. I'll talk about collaborations. I'll get credit to talk about collaborations. So, given this history, my kids are starting, get the ancestry stuff. I didn't do it. It's okay, fine. I can figure it out from what you guys have. And so my wife came back. She's 34% European ancestry. And then my son and my daughter, their stuff came back, and they were 24% European ancestry. So I'm very proud of the fact that my kids can do math. So we're at the dinner table. They're looking. I said, "Well, you look at me for." My wife said, "Well, who else are we going to look at?" So, I'm going to go ahead and take the test, and we'll find out, what's new. I'm not going to. I don't think my haplotype will be as colorful as Lizo's, but we'll find out what it is. Okay, so I'll stop here, and then I want to talk a little bit about collaborations, multidisciplinary collaborations, what you have to do, how to use communication, and how we try to work through things. And a lot of the work that I showed today was the computation stuff was done by Sandeep Singhal. He's right there. You want to give him credit for it. But this is a big project that goes across multiple different universities and institutions, and it's not over. It's going to continue for quite some time, for the next couple of decades, I hope. So, and I want to acknowledge the funding sources for intramural research program, from NCI for program project grants, and we have commercial funding from Ultiview, which is doing all the fluorescence for us. Now, my last thing, I just want to talk about multidisciplinary collaboration, just give you some advice and give you some insight from our experience, okay? I'm a pathologist. We're working on a project that requires multiple different people from multiple different backgrounds. We're used to the idea of the consult. So we like to get advice from different people, but it becomes different when we start bringing in the data scientists, computational sciences, image analysis, scope. And so what I do, everybody gets in the same room. We all really don't speak the same language. And I began with a lecture. I said, look, there is not, the academic enterprise is robust and resilient. There's nothing anybody's going to say in this room that's going to break it, okay? And we're here to teach each other. We're not here to judge you because you don't know enough about our field. Because if I don't know enough about your field, I'm glad you're here, okay? And so that's how we talk to each other. But it's still pretty tough. We started this. We have this bright kid who's his background is in particle physics. We have a director, his background is in particle physics. We begin talking, and he starts giving a talk, and I'm listening. I said, well, I know I only speak one language, but now I feel that I don't even do that because Andy, when you said there is, I understood that, and then everything after that, I had no idea what you were saying. And so we really have to learn how to slow down and think of how we're communicating. That's the only way it's going to work. So, my advice is we have to learn to communicate, okay? And although we may start out like this, in the end, we kind of end up like this. And that's the end. Thank you very much.